Dear students, today I am going to deliver the lecture on tumor immunology, immunodiagnosis, and immunotherapy of cancer. First, I will start with tumor immunology. Tumor immunology has been defined as part of immunology that deals with the antigens on tumor cells and the immune response to them. As a consequence of their loss of differentiation, tumor cells may express developmental antigens that are usually seen only in the uh, prenatal period. Examples of these antigens are alpha fetoprotein and carcinoembryonic antigen. Tumors or neoplasia is said to develop when the balance between cell death and renewal is distributed in a way that numerous clones of a single cell group are produced in an uncontrolled fashion. It has been postulated that the immune system is responsible in part for the protection of the body against the development of malignancies. At the same time, the prevalence of numerous cancers in immunocompetent individuals indicate that immune system has only a partial role in protecting against malignancies and also that it is not very efficient at it. So tumors induced by some virus that express antigens shared between different tumors. And these consist of membrane expressed virally encoded antigens and have been termed as tumor associated transplantation antigens. Since they are not strictly speaking as tumor specific, so there are two types of antigens. One is two groups of antigens. One is tumor specific group, tumor specific transplantation antigen, TSTA, and tumor associated transplantation antigens or THA. The third one is that is the oncofetal antigen. So tumor specific antigens, the first group TSTA, is called are the unique to tumors. They are not found on other cells of the body. They are usually the products of mutated genes seen in the cancer cells. Cytosolic processing of the abnormal proteins yields peptides that are unique, and when presented by the appropriate MHC class 1 molecules, it elicits cell-mediated immune response. Various physical and chemical carcinogens cause malignancies by inducing mutation in key genes involved in modulating cell growth. One of the proto-oncogene, that is RAS, including the P21 RAS protein and other related gene products, are an example of this tumor specific antigen. RAS proteins bind guanidine nucleotide GTP and GDP and causes intrinsic GTPase activities. The mutation associated with RAS genes in malignant cells appear to cause a single amino acid substitution at specific position 12 or 13 or 61, which results in increased enzymatic activity of the gene product. As a consequence, the cell acquires transforming capacity. So different virus-associated cancer or tumor-specific genes are there, and the viruses that have been implicated in tumorigenesis include Epstein-Barr virus, hepatitis B virus, hepatitis C virus, etc. The second one, that is the tumor-associated transplantation antigen. These are the other class of tumor antigens, and these antigens are expressed by the tumor cells and also by normal cells at low level or only during the progress of differentiation. The expression of these antigens is considerably sorry, uh, de-repressed or enhanced after the process of malignant transformation. So, Tatas are of different types. There may be tumor-associated carbohydrate antigen, differential antigen, and the third part, that is oncofetal antigen. So these oncofetal antigens are found in embryonic and malignant cells but are absent in normal adult cells. So these are TATAs which are more or less selectively expressed on tumors. Alpha-fetoprotein and carcinoembryonic antigens are examples of these antigens, which are found in uh, hepatomas and colonic cancers. Silent tumor-associated genes are not expressed in normal cells but are actively transcribed in tumor cells. Tissue-specific genes or differentiation genes are present in the surface of normal cells or may be said to the circulation, but the levels of expression are usually very low in normal cells. Examples are carcinoembryonic antigens or alpha-fetoprotein, as I said. So, this, so the tumors, they are stimulating the immune response in our body. And tumor antigens are capable of eliciting a comprehensive immune response involving both the cellular as well as humoral immune responses. In case of cellular immune responses, T lymphocytes play an important role in tumor immunity. They act both as cytotoxic effector cells and as central modulating cells. Through these effector cells, they control the specific cell-mediated anti-tumor immune responses and upregulate non-specific killing mechanisms. The activation of the T lymphocytes by tumor cell products as a consequence of antigen recognition may result in the secretion of non-specific immunoregulatory factors. And these factors are capable of regulating the tumor killing functions of the mononuclear phagocytes, natural killer cells, granulocytes. And these factors also enhance the ability of the natural killer cell and monocyte to participate in ADCC against tumor cells. They can also include the macrophages. So macrophages also play an important role in tumor response. Clustering of the macrophages around tumor cells is associated with tumor regression and seen in the cases of numerous cancers. Humoral immune responses, the second type, here the B lymphocytes produce tumor-specific antibodies which may induce complement-dependent cytotoxicity of tumor cells or may mediate ADCC. ADCC can be mediated by a variety of cells expressing the Fc gamma receptors of the NK cells, monocytes, or macrophages, or granulocytes by recognizing and destroying immunoglobulin G coated tumor cells. So this is all about tumor immunology. Now I'm coming to immunodiagnosis of cancer. A number of immunodiagnostic procedures in cancer is clinically important in the sense that the tests may confirm the result of other cancer diagnostic investigations. However, each one of the tests has its own area of application. Immunodiagnostic tests are used for the detection of relapse or metastasis as well as for the histological verification of the tumor. A mass screening for tumors in the population and most probably the early tumor diagnosis by immunological means is impossible due to biological variables among the tumor bearers and to technical difficulties. Immunotherapy of tumors is still at an experimental stage, although immunization is possible to animal tumors. Convincing data on similar effects on the human tumor system do not exist as much. There are different antigens has been identified, and these antigens has been targeted. And if we can identify these antigens, and if we can check the status of the antigen, then we can diagnose that how much immunological mechanism occurs under these conditions, or what is the probability of the development of a specific tumor or specific cancer in the individual. So there are different type of approaches, mostly on the specific antigens, and one of the specific antigen is prostate specific antigen. It is a glycoprotein located in ductal epithelial cell of the prostate gland that can be detected in low concentration in the sera of healthy men. Using an appropriate upper limit of normal assays with monoclonal antibodies detect elevated serum level of PSA in about 90% of patients. So the antigen that is present in serum, the assay of the serum PSA level is very useful marker for prostate cancer, perhaps the most meaningful serum marker for neoplasia. In healthy men, the level of PSA vary in between zero point 0.6 to at the age group of 21 to 30 and 1.5 nanogram per ml at the age group of 60 to 70. But under the condition of prostate cancer, it is significantly elevated. So PSA can be used to monitor prostate cancer conditions as well as recurrence after prostatic carcinoma has been diagnosed and treated. Next one is CA125. CA125 is clinically useful for the screening of and diagnosis and also monitoring the therapy of ovarian cancer. Although any peritoneal inflammatory process and some other cancers can also increase the level of CA125, but it is specific to ovarian cancer. Beta 2 globulin is often elevated in multiple myeloma and in some lymphomas. So its primary use is in the prognosis. The other one is CA19-9, was originally developed to detect colorectal cancer but proved more sensitive for pancreatic cancer. It is primarily used to judge the response to treatment in patients with advanced pancreatic cancers. And this particular protein can also be elevated in other GI cancers, gastrointestinal cancer, particularly cancer of the bile duct and some benign duct and cholestatic disorders. Means this is cholestatic disorder means where the bile secretion is not sufficient, it is obstructed. So these are the method we can use for the diagnosis of different type of cancer. Now I'm coming to cancer immunotherapy. So the immune system has a natural capacity to control the development of cancer, which is known as cancer immunosurveillance. During tumorigenesis, a process in which normal cells become malignant, tumor cells accumulate numerous mutations in DNA. This leads to production of abnormal proteins, the so-called neoantigens or tumor-specific antigens, which are perceived as foreign protein by the immune system. Neoantigens trigger the secretion and expansion of tumor-specific T cells, which are able to recognize and kill tumor cells. However, developing tumor induce several mechanisms to avoid recognition and effective destruction by immune cells. Cancer immunotherapy is a type of cancer treatment which aims to induce natural defenses to fight cancer. It involves the use of various substances including small molecule drugs, cytokines, monoclonal antibodies, as well as immune cells which trigger and enhance or restore the body's own anti-tumor immune response. So the types of cancer immunotherapy there are four types: monoclonal antibodies, it may be conjugated or unconjugated form, cytokines, adoptive cell therapy, and cancer vaccines. First, I will start with monoclonal antibodies. MABs are immunoglobulin produced in the laboratory that specifically recognize and destroy the cancer cells. The variable regions of anti-cancer monoclonal antibodies specifically recognizes antigens which are exclusively or predominantly localized on the surface of tumor cells. Like natural immunoglobulins, therapeutic monoclonal antibodies destroy tumor cells by a number of mechanisms which engage other components of the immune system. They can activate specific protein cascade, which is known as the complementary pathway, which results in direct cancer cell killing and stimulation of inflammatory reaction. In addition, monoclonal antibodies bound on the surface of tumor cells engage binding of immune effector cells with the ability to exert cytotoxic effects. This clinically relevant mechanism is called ADCC, antibody-dependent cell-mediated cytotoxicity. Some of the therapeutic monoclonal antibodies can also directly induce apoptosis in target cancer cells. There are different approaches. So we can have antibodies that is tumor specific. It will adhere to or it will adsorb to the tumor cell with a particular receptor. Here you can see that CD16 receptor. So NK cells with their X receptor, that is the CD16, are activated to kill the tumor cells. Next, tumor specific antibody or antibody fragment conjugated to a toxin. So this antibody toxin conjugate bind to the tumor cell and the conjugates are internalized within the tumor cell and killing it. The third group is the tumor-specific antibody or the antibody fragment that is conjugated with a radionuclide, which is known as radioimmunotherapy, RIT. So here the radioactive probe that is conjugated with the monoclonal antibody binds to the tumor cell, and radiation of the tumor cell kills the radiation coming from the radioisotope is killing the tumor cell. And here also, not only this is specific, they are also killing the surrounding normal cells also, tumor cells as well as the normal cells also. So these are the methods conjugated monoclonal antibody we can use, either in the form of radioimmunotherapy or in the form of conjugated chemotherapeutic molecule or chemo-labeled. Next is the cytokines. So cytokines are a large group of proteins. They are antigen non-specific molecules or immunotherapeutics that are boosting the immune system in a general way. They involve substances that stimulate production of cytokines. So these are proteins produced by the immune system to control immune cell development, function, and migration. Currently, recombinant cytokines are produced on a big scale in cell culture system. Some cytokines are interferon alpha, interleukin 2. So these are the most common type of cytokines. Interleukin 2 helps immune system cells to grow, to divide more quickly. And also IL-2 can be used as a single treatment for cancer or can be combined with a chemotherapeutic drug or with other cytokines such as interferon alpha. The second one is the interferons. They are used to treat different type of cancers, like here is the leukemia, chronic myeloid leukemia, follicular non-Hodgkin's lymphoma, cutaneous T-cell lymphoma, kidney cancer, melanoma, Kaposi sarcoma. So a wide variety of cancers can be treated using interferons. So the third type is adoptive cell therapy. So adoptive cell therapy is a form of cancer immune therapy which involves the collection of patient's immune cells from blood or tumor tissue, their subsequent activation or modification to increase recognition and cytotoxicity towards the tumor cells, followed by injection back to patient circulation. Historically, the first type of adoptive cell transfer involved tumor infiltration lymphocytes, TILs. So T cells infiltrating the tumor were collected from tumor tissues, expanded in the laboratory, activated by the treatment with immune stimulating cytokines, and re-injected to the patient's bloodstream. Now the T cells along with the NK cells are the immune cells that are most frequently used in adoptive cell transfer. So the reduced number of injected T cells is accompanied by the interleukins, IL-2, and they are reducing the side effect of this adoptive cell therapy. So interleukin 2 is normally added to the extracted T cells to boost their effectiveness, but in high doses they can be toxic. So we are not using in high dose. So reduced number of T cells, reduced number of IL-2 will be accompanied and they are re-injected into the patient's bloodstream. And under that conditions, they are reducing the toxic effect or side effect of the interleukins. So this is the scheme of adoptive cell therapy. So here, this is targeted therapy using one monoclonal antibody, hemorrhaginib. So this is pro-apoptotic signal, dying tumor cells. Now we are taking adoptive cell transfer. So here is your tumor antigens. Now what we are doing? We are recruiting and activation of the adoptively transferred T cells along with the endogenous T cells and interleukin 2 here, boosting the antigen presenting cells, and along with these cytokines, with chemokines, anti-angiogenic factors. So what they are doing? They are interacting with this tumor antigen, tumor specific antigen, and they are resisting proliferation of the tumor cells. They are killing the tumor cells. So this particular process is known as adoptive cell therapy. The last one is cancer vaccine. In general, a vaccine is a preparation of an antigen that drives immune response and provides acquired immunity to a particular disease. There are two types of cancer vaccine. One is the preventive cancer vaccine and the treatment cancer vaccine. So it is recognized that some cancer types are caused by microbial infections. Say human papilloma virus can directly contribute to cervical cancer, anal cancer, orthopharyngeal cancer. Similar to infectious diseases, preventive vaccines contain microbial antigens and prevent from a viral or bacterial infections, which indirectly protects from cancer development. In contrast, therapeutic vaccines, these are anti-cancer vaccines which are given to a tumor-bearing individual, not before, during the treatment. They contain patient's own inactivated tumor cells. And this inactivation of tumor cells will be done by irradiation or tumor-derived antigens and stimulate the development of a specific anti-tumor immune response. Many different variants of anti-cancer vaccines are investigated already for the decades. However, so far therapeutic vaccines are not widely used in the clinics. So there are some limitations of immunotherapy, mostly monoclonal antibody mediated. They are not administered as a first-line therapy, second, third, or maybe the last hope, last resort cancer treatment. When the immune system is already weakened by chemotherapy, surgery, as well as radiation, we can use this therapy. Monoclonal antibody-based immunotherapy. It is conjugated with radioisotope or conjugated with chemotherapeutic drugs. Second one is that not all antigens are same. Because all cancers looks like they are similar. They are same, but they are not. Not all patients' cancer may express the antigen against which a specific monoclonal antibody is targeted. In general, response rate of these targeted therapies appears to be 20 to 30% only. Because there is a variety of their surface antigen. And patient to patient, they vary. The third one is that tumor cells mutate. So if the tumor cells of the cancer cells, they are undergoing radiotherapy or chemotherapy treatment, they will change, they will mutate themselves. And under such conditions, if you use immunotherapy, it will not work because the target has changed. The monoclonal antibody has a single binding site that is specific towards an antigen, could not attach with it and could become ineffective. And the last one is toxicity. So toxicity is associated with some targeted therapies and which is significant. So we cannot use it because toxic side effect is significant amount. So these are the limitations of immunotherapy in cancers. That's